Morning, everybody. Dr. Gillard here. It is week 7. Can you believe it? Week 7. It is Monday, week 7, spring 2020, and this is our 7th, really our 6th embryology lecture. Here we go. It's an important one today. We finally finished the midterm. We finished the week of 2s, and now we're starting week 3. A lot of busy stuff in this week. Really important stuff. Obviously I can't go through, in this tiny class, I can't go through all embryology. You're going to have to do some board studying yourself on this. But if you understand what we've taught you so far and what I'm going to teach you, you'll be able to easily understand how other things are made, because this is the root of everything we're going through, kind of the root of everything. All right, we're making the germ layers, and then we're going to fold it. Fold the disc up, and from there you can take it from there. All right, in general, the week of 3, the star of the show is Adele. It's not Adele, but probably the best voice I think I've ever heard. She's got a great voice. But anyway, the star of week 3 is not really Adele. It's gastrulation. It's a new word for you, gastrulation. Week 3 also marks the start of the embryonic period. The embryonic period. The embryo is really going to develop. We're going to form organs. Therefore, an AKA for week 3 is organogenesis begins. The embryonic period is week 3 through 8, so it's a big period, but we're just starting off here. And gastrulation kicks off the show. So it starts on day 14. That's the start of week 3. Right at the beginning, it's the most important event of week 3. Marks the beginning of morphogenesis. That's another one you could say. So I could have added that in here, embryonic period. Organogenesis. Another one is morphogenesis. That's another chiropractic board book. So it's another kind of AKA for this period. And what is the definition of gastrulation? It's the process of converting our bilaminar disc. Remember, we only have an epiblast and hypoblast right now. That's all you know. We're going to convert it into a 3-layer disc. And then every single one of those cells is going to be morphed into a stem cell-like creature. And those are going to be called the germ layers. We're going to create the 3 germ layers. So we're going to make a new layer. In fact, you can see, there's what we have right now. We have an epiblast and hypoblast. But we're going to create a brand new layer in the middle. That's going to be called the mesoderm, or intraembryonic mesoderm more specifically. And then we're going to convert this into an ectoderm and this into an endoderm. That's where we're going. Before we can do that, we have to form a new structure. We got something new here. That's the primitive streak. And that's got to happen first. Remember, we kind of introduced you to that already, because we have to make sure that doesn't form where? Cranially. It's going to grow cranially, but it forms caudally. Right? Cranially would be right up here. All right. Okay, forming the trilaminar disc. So after the primitive streak is mature, we're going to convert the epiblast and hypoblast, everything I just said, into 3 new layers. These are called the germ layers. There's a embryonic ectoderm. Ectoderm is what most people call it. There is a intraembryonic mesoderm, usually just called mesoderm, but really you have to be careful because we studied extraembryonic mesoderm, didn't we? Right? We studied that. So this is intraembryonic mesoderm. And then we're going to make an endoderm. The ectoderm used to be the epiblast layer, and the endoderm used to be the hypoblast layer. And we're going to get rid of those layers now. We're changing the cells. In fact, some of these cells, these hypoblast cells, are going to get kicked right out of the area. some fun facts. Before you can make the 3 germ layers, can't do that without making a primitive streak. Right? Primitive streak will set up magic cells that induce cells passing through them and tell them what to do, as we'll see. It's kind of like a boss layer or a boss streak. It tells the slaves where to go, what to do. Primitive streak is the biggest event of a very busy week 3. Lots of stuff there's lots of stories going on simultaneously. We'll kind of come back to some of those, like the formation of the prechordal plate is going to be starting kind of at the same time, and it's a lot of stuff going on. Oh, there's our little housekeeper. We got some housekeeping to do. So we've been talking, remember we went from a morula to a blastocyst. Blastocyst stayed with us through week 2, and now we're going to change the blastocyst. You don't call it a blastocyst anymore. You call it a gastrula. Gastrula. And if you're in doubt or you can't remember, just call it an embryo, because remember the embryo has been with us for a very long time, right after the zygote phase. You could still really call it a zygote if you wanted, but real star of the show is the epiblast. These epiblast cells, remember those guys? This layer, this gray layer, well, they've already converted into their germ layer into ectoderm. But the epiblast layer, those are super potent stem cells. There's only one kind of stem cell that can morph into any tissue, from a brain cell to a toenail cell to a heart cell to the lung cell to your tooth cell. These stem cells are called pluripotent stem cells. Pluri means they can go anywhere. They're amazing, amazing cells. Pluripotent stem cells. And these pluripotent stem cells literally create every single layer, every germ layer, its father or its mother, its beginning is the epiblast cells. They're magical. If we could harvest those and use those for research, of course we can't. I won't go down that road, but boy, we're just missing out by not being able to harvest those. We could do some incredible things with them. Grow a new liver, grow a new pancreas, grow a new lung. So much we could do with them. All right, back to Adele, gastrulation, the formation of the primitive streak. All right, so that's the first step. What's the first step of gastrulation? Forming the germ layers? Nope. Can't form the germ layers until you form the primitive streak. So primitive streak is a super important signaling center. These cells are going to have all sorts of genes turned on, and they're going to be able to induce cells that pass next to them. In other words, they're going to flick switches in cells passing through them, and basically are going to instruct those cells where to go and what to become. So it's important. We'll look at the primitive node. That's even more important. That's like the aircraft, or that's like a control, an airport control tower. That's got some even more important genes in it. Then we'll look at the primitive pit and primitive groove here in a second. All right, remember the first thing, this is where we started. This is what we have right now at the end of the second week. We have a bilaminar disc, 2 layers. We talked a lot about these AVE cells, and they're not really that big. They're the same as these little yellow ones here. That's the hypoblast. The blues, the epiblast cells, more columnar in shape. Amniotic cavity, secondary yolk sac. Extraembryonic coelom or chorionic cavity is out here. Remember all that stuff? We have this, what's this inner layer called? Splanchnic, extraembryonic mesoderm. And this somatic extraembryonic mesoderm. All right. So our goal is to turn that into a 3-lam. Our goal is to turn that into that. That's what we're working on. All right. Now, remember nodal. We talked about the gene nodal and its gene product, how important it was. Remember that the AVE cells went to great lengths to make sure nodal was turned off in itself and in the cells above. Remember, nodal, we don't want that there because nodal induces the formation of the primitive streak. So you don't want that starting up at the cranial end of the bilaminar disc. It has to start at the caudal end. Right? Only caudal epiblast cells have the power to form a primitive streak. So recall, nodal in the cranial region secretes the nodal inhibitors. The mnemonic could be cold, CLD, Cerebrus 1, Lefty 1, DDK 1. Those are the nodal turner-offers. And yeah, those signaling molecules diffuse into the cranioepiblast cells and turn off the nodal gene. Without nodal, the cranial epiblast can't form a primitive streak. That's real important. You get horrible mutations if that doesn't occur. What else? We got another system that stops primitive streak formation. AVE cells also secrete something called noggin, and noggin blocks the BMP4 gene. And without BMP4, the caudal structures like the streak are prevented from forming cranially. So noggin gene is important to be turned on as well. So you got these nodal turner-offer genes, the cold, Cerebrus 1, Lefty 1, DDK 1, and you need the noggin gene turned on as well. Okay, induction of the epiblast. Let's start the process. So how do we make the 3 germ layers? The very first thing you have to do is you have to tell the epiblast cells, the ones that are way out on the lateral margins of the disc, you have to tell them to migrate and migrate toward the midline. And so you get a big migration of these laterally positioned epiblast cells, and they move toward the middle and they start piling up in the middle. And this creates a line, a structure, and that's the primitive streak. Okay? The peripherally located epiblast cells are said to be induced, and guess who induces them? Amongst other things, nodal, the nodal gene product. Because remember, we're in the caudal end, and nodal is running wild there. All right? So nodal induces them to, and we'll see there's a little more to the story because they have CAMs, they have cell adhesion molecules, so they can't migrate anywhere yet. But in general, we got to get them to move toward the midline. And again, that midline, that movement only occurs in the caudal region, right? Because that's where nodal is turned off. It doesn't occur in the cranial region because, or it occurs in the caudal region because nodal is turned on. It doesn't occur in the cranial region because nodal is turned off. Right? So here's a kind of a blow-up of that picture we're looking at. Here's our embryonic disc. Here's the dorsal surface up here. Ventral surface would be down here. And a bunch of cells, I guess I could have drawn this a little better, but a bunch of cells get stimulated by nodal, and they start moving. They start conveyor belting right in toward the middle, and they start piling up. And this pile of cells is the primitive streak. Okay? So some epiblast cells are migrating toward the midline and piling up. And now once the primitive streak is formed, you don't need electron microscope anymore. You can see this. You can tell which way you get the caudal cranial axis. So we know the caudal is back here. Cranial is here. We actually know where left and right is now too, right? So this would be the left side, left part. This is going to be the right side. So we have some axes established. Okay, and again, how come the streak didn't form here? No nodal or no BMP4. Need those if you want to make the primitive streak. All right, now back up a little. What causes these cells to migrate in? How do they know? Who induces them? What's that mean? Who induces them to move. That's like, who tells them to move? Well, it's a multi-step process, but the first thing that happens is the WNT3 gene turns on the nodal genes. I kind of said nodal already, but if you want to get specific, it's you got to have the WNT3 gene on first, and that turns on the nodal gene. Nodal stimulates then the lateral epiblast cells to migrate, tells them, hey, get out of here. Go, you're lucky you got chosen to be a primitive streak cell. So you need to migrate toward the midline. So we can say that these gene products stimulate or induce the midline migration of epiblast cells. And we're going to back up, because we got to do some more work on these epiblast cells. Run-of-the-mill epiblast cells can't go anywhere. Once the epiblast cells do pile up in the midline, we said, then they keep coming in. So where are they going to go? They're actually pushed forward toward the cranial end, right? And as they pile up, so the primitive streak grows cranially, and because they pile up and grow cranial. And once it gets about halfway toward the ultimate cranial end, it stops. So the primitive streak only occupies about 1/2 of the dorsal caudal surface in the midline of the embryonic disc. Okay, and so it's reached the halfway point, but cells are still coming up. So what's going to happen? The cells actually start piling up at the cranial-most end, and they form like a little knot, right? So here's the primitive streak growing like this direction. More cells come in. Now they start piling up. How come it doesn't keep growing? Why doesn't it grow up into this region? Well, remember the nodal's turned off here. So they can't grow any further. These cells pile up and become the primitive node, or primitive knot, or Henson's node, or the organizer. It's a board book. Langman calls it the organizer. Why don't they should just call it the primitive node. I don't like when Langman goes off like that. Makes it difficult. Okay, here's a overhead view. Which way's cradle, which way's caudal? This is the anterior. Sometimes some authors call this anterior. How in the heck can anterior and cranial be the same? Because this is going to fold, and when it folds, the cranial is going to be folded into the anterior direction. So some authors try to confuse you more by calling it anterior, but just watch out for that. At this stage, they're AKAs. Here's the primitive streak. And there's the primitive node. It said it doesn't grow much more than halfway. That primitive node is super, super important. It is a major, major signaling center. All sorts of genes are turned on there. It can really instruct. We're going to see it instruct cells to make the brain and amongst other things. So it's really, really important. It's said to be a major signaling center, kind of like an airport traffic control tower, organizing, telling planes when to land, when to take off, go in a holding pattern. Just organizes everything. Oh, there's the little nerdy guy. Here we go. We got some more genes to learn. Aren't you excited? These are kind of cool names, though. Look at that goosecoid, FOXA2, chordin. So these are the three classic genes turned on in the primitive node. Goosecoid and chordin, for that matter, are both really important in neural induction, forming the neural plate and neural tube, and we'll come back to them. Goosecoid activates noggin and chordin, which are important for creating symmetry of the body. How come the heart knows to go on the left side? Because the cells that form it have the goosecoid gene turned on. Goosecoid activates noggin and chordin, and that creates the sidedness. And the chordin is turned on by goosecoid. It's also important in neural induction. FOXA2, what does that do? It helps the primitive node mature, so it's important for that. It's vital for proper formation and structure of cranial of cells going further, which are going to be like the prechordal plate. Well, vital for proper formation of structures, cranial to the node. Yeah, so cells are going to migrate through that primitive node, and the FOXA2 gene needs to be on to tell them, for example, to become prechordal plate. It's important for that. And super important, we'll talk about next time, the nodal cord. So it's mandatory that gene has to be working, or you're not going to form a nodal cord, and you won't. If you don't form a nodal cord, you're going to have trouble. Downward and forward migration. So epiblast cells will not only migrate into the primitive streak, they keep right on going down into the epiblast. Actually, there's a space between the epiblast and hypoblast. They're going to migrate and kind of jam into that little space to create a new layer. Right? They'll also migrate forward in the midline to form some things like the prechordal plate, amongst other things. The nodal cord itself is another migrating structure. Kind of a cool little cartoon here. So we have pretty much where we are. We got our purple, that's the primitive streak that's formed. Let's blow it up and look at it here. Well, let's look at the parts. So we got primitive streak, hypoblast. Now we got a new space, don't we? These slippery little cells are kind of slipping their way in and making a new space. That's going to be, what is that going to be? That's going to be the mesoderm. All right? Epiblast is in blue. AVE cells are in red. Hypoblast is here. And you can see we got all these epiblast cells. They're really going to be called epiblast cells turn into flask cells, which turn into mesenchymal cells. So these are really mesenchymal cells that can swim. They have pseudopodia. They can literally move and swim. So all these cells can migrate. And we have cells migrating through all areas of the primitive streak and getting underneath between the two layers here, the two germ layers. I was going to say endoderm and ectoderm, but it's not yet. It's still hypoblast and epiblast. So they're squeezing in here. We got these special cells kind of heading up here. These are going to form the prechordal plate here in a second. Then they're going to form the nodal cord, the second wave. The first wave of, because we're in the mid-sagittal cut here, the first wave is going to form something right up in here. It's called the prechordal plate. Then the second wave will form the nodal cord. Okay, as cells start pouring through and going through the cells of the primitive streak down beneath, they start carving it out. And so you get what's called a primitive groove. And they also go through the primitive node so much that it creates a hole called the primitive pit. Right, so there's an embryonic disc. We can see we got mesoderm starting to form here, but we got, it doesn't show you, these little cells moving in here, going down, and then they squeeze and push apart the epiblast and hypoblast. That's going to be mesoderm there. But you can see the point of this is to see the primitive groove and primitive pit. There's an electron microscope of this. You can see the primitive groove quite nicely. Here's the primitive node. I mean, this whole area would be primitive node. Primitive pit would be a hole in there you can't really see. All right, so now we got body axes formed. We kind of talked about this already. We know dorsal, we know ventral, we know cranial, we know caudal, we know left, we know right. Guess I could probably take these slides out. All right, now that the primitive streak is formed, yay, are we ready to make the germ layers? No, we got a little bit of work to do, and we actually have to back up a little bit. The epiblast cells can't do anything yet because they're still joined together. Right? So let's back up. Epiblast cells are just columnar epithelial cells, like the run-of-the-mills, and they have really strong attachments between the cells that need to be broken. Those are called CAMs, cell adhesion molecules, or junctional complexes. Either one of those things. They need to be broken. So let's see how that happens. Here's the CAMs for histology. You better know these, right? Tight junctions, these are the ones that stop. If this is the GI tract and there's a bugs here, the bugs try to get between the cells and they can't because these tight junctions keep bugs, keep acid, keep things out. They're pretty good at that. They're terrible at holding the cells together, though. If you pull the cells, you could rip them right apart. But these guys, zona adherens and macula adherens, these guys hold these cells together, especially these macula adherens, which are also more commonly known as desmosomes. So they actually, you can see them binding to the cytoskeleton, and so they hold it together really, really strong. But we need to break those loose, right? So warning, high area of author confusion. Authors are all over the place on this. There's just not enough human embryos to study. So we're studying mouse embryos and chick embryos and reptile embryos, and that's where all this story came from. So we need to break those chains. That was a great, I don't know about the last season, but that was a really good series. Probably the best one I think I've ever seen. But preparing for ingression, the breaker of chains. All right, so epiblasts are locked together, as we said. They have two types of junctional complexes that need to be broken. Without them breaking, they're not free to migrate anywhere. We already said zona adherens and macula adherens, desmosomes. What are they made of? What actually holds them together? Things called cadherins, proteins called cadherins. These are desmoglines and desmocolons. Those are specific members of the cadherin family. I think probably if you knew cadherins, that'll be good enough. You could ask Dr. Doe on that one, how deep you need to go. So these extracellular portions of the junctional complexes are just proteins of the cadherin family. So we need to break cadherins in order to set them free. And that's exactly what happens. Epiblast cells that are destined to migrate, they produce certain proteins, one called snail in particular, that turns off the genes that makes cadherins. Simple as that. Turn on snail, no more cadherins. Cells get really loose. And that's what we need, right? So snails, snail is the freer of the epiblast. That is a snail. Don't forget that snail. So the snail gene, breaking the cadherins, is accomplished by a transcription factor, which turns on other stuff we won't worry about. But snail is really important. That gene has to be turned on in the epiblasts that want to migrate. Snail will turn off the genes, decrease the expression of the genes that make cadherins. And therefore, if you give them a little time, those cadherins have to be replaced all the time. You won't be able to replace them because that gene doesn't even work anymore. All right, so it frees the epiblast layer. All those cells are free and ready to migrate now. Okay, but we're not quite ready to migrate because there's really nowhere to migrate. We have to migrate through a very slippery or a very tight space. So we should make it slippery, shouldn't we? That's exactly what happens next. So our epiblast cells that are destined to become travelers or migrators, not only do their snail is created to break the cadherins, but other genes that turn on hyaluronan or hyaluronic acid are turned on. And so these cells start spitting out hyaluronic acid, which is like snot, right? It's really slippery. And that's what it does. Some of the hyaluronic acid actually sinks down underneath the epiblast, and it gets in between the epiblast and hypoblast and starts making that slippery. We're going to need to slide cells into that area, so it's a good thing to make it slippery. And it also coats itself, so it makes itself slippery so it can slip into the space between epiblast and hypoblast. There's another gene, the fibronectin gene, is also turned on, which is slippery. So that's another one. Hyaluronic acid gene on, fibronectin gene on. If you're going to be a migrator, it has to be on. Now let's modify them. So now we have the epiblast cells broken. They're free of their neighbors. They're slippery. Are they ready to go and become mesoderm? Are they ready to dive through the primitive streak and become mesoderm? No, not yet. Well, I mean, they are. But they have to do some morphing, because how are you going to crawl down to the hypoblast? They don't have anything to crawl with. They're just columnar cells. So they morph into a new cell type, and this is called a bottle cell. The bottle cell is the one that actually passes through the primitive streak, and it's changing. By the time it gets through the primitive streak, it turns into a mesenchymal cell. And mesenchymal cells, you might remember from your biology, they have these like weird little hands, right? And they can use those hands to move. And that's exactly what they do. Here's a cartoon. So epiblasts are moving this way, and the cams are broken here. They're getting slippery. And as they start passing through the primitive streak, they're transformed in this bottle cell. And the bottle cell is converted into a mesenchymal cell. And that can go anywhere. See, it's got the little pseudopodia, these little arms, so it can crawl, it can do whatever it needs to do. All right, another thing has to happen. So epithelial, because remember I said they're simple columnar. Well, now they're bottle cells, now they're mesenchymal cells, so they're not epithelial cells anymore. So in order to be a traveler, a morpher, or a cell that migrates, you actually have to turn into mesenchyme. So there's a process for everything we just described about going from an epithelial cell to a bottle cell to a mesenchymal cell with arms, and that's simply called epithelial to mesenchymal transformation, right? EMT, in other words, epithelial to mesenchymal transformation. Are we good with that? We're going to talk about EMT when we talk about creating neural crest cells, which are really important. Now, what can they do? Oh, now that they can migrate, how do they know where to go? Okay, so now they're slippery. They have grown arms. They're mesenchymal. We got the space between the epiblast and hypoblast all greased up, right? But now how do they know where to go? I mean, the committee programs them, some believe, but there's evidence that there's two main genes turned on that tell them where to go. So it's these two main genes in the primitive streak that tell them or direct them what to do. So, of course, you want to meet them, right? Should have put more stars on this slide. I've asked this before. We have the brachyury gene, pronunciation, brack, like back, your, brachyury gene. And that is the major category in the T-box family of transcription factors. So it's a transcription factor, stimulates DNA to make something. So brachyury activates FOXA2 and goosecoid. Remember, we talked about them already. So how do they get turned on? It's brachyury gene is turned on. They also, we won't get into the mechanism, but they protect the bottle cells as they go through the primitive streak. That's important. And then last but not least, they help induce the formation of the nodal cord. All right, we'll talk about that next week. It's super important, that nodal cord. So brachyury gene, another one that's turned on is nodal. Well, we already know nodal's turned on, but it's actually turned on at different magnitudes in different regions. So cells that get exposed to high levels of nodal actually become endoderm. Cells that are exposed to low levels of nodal, as they pass through the streak, right? All the streak cells are secreting nodal, but some secrete nodal really high. So there's the primitive streak, there's the node, and we got these cells passing through. As they go through, they're getting doses of nodal, and the ones up here get very high doses of nodal. The ones back here, they get kind of low doses of nodal. So nodal, the dosing of nodal tells the cells where to go. If you get super high levels of nodal, I always think you're going to have enough power. Think of nodal as like energy. If they get a high dose of energy, they'll have the power to go all the way down to the endoderm or to the hypoblast cells, kick those hypoblast cells out, and become endoderm. If they experience low level or they're exposed to just low levels of nodal, then they don't have power to go all the way down. They have power just to go underneath the epiblast, and they become mesoderm. Are epiblast cells ready to migrate now? Yeah, they're definitely ready, right? They're free of junctional complexes. The space is lubed up with the gags. And yeah, we've already made mesenchymal cells. So the prepared epiblasts now migrate downwards through all regions of the primitive streak and the primitive node. This process is called ingression. Ingression. Langman calls it invagination, board book. So other board book, more chiropractic board book. So the word ingression could be on the boards. Langman, chiropractic board book. Invagination could also be in the boards. You got to know them both. Here's a cool little picture. So yeah, there's the primitive streak. We have all our little mesenchymal cells. They were hypoblasts, then they became bottle cells. Now they're mesenchymal cells, and we can see them coming down through all regions of the primitive streak in this mid-sagittal cut. And they've actually pushed apart a region, and this is mesoderm layer. Okay, we also have some going through the node, primitive node. These guys are in the midline here. These guys are going straight up here, and they're going to form something called the prechordal plate up here next. That's that first wave of migrating cells does that. All right, mesoderm first. So this is kind of arguable. Some books say the endoderm forms. Some books say mesoderm forms. Probably in reality they form at the same time. But here we can see all these cells loose, moving in. They're slippery. We see them dropping down through the streak, and they're going to different regions. Some are hanging out here to produce mesoderm. Some are going all the way down to the bottom and knocking those old hypoblast cells right out of there. Those hypoblast cells will be knocked right out. Maybe they'll make a little extra embryonic mesoderm, but we're going to form a new layer. That's going to be the endoderm layer right there. Right, and here's just as I said, so we got some of the cells have been kicked out here, form extra embryonic mesoderm, and we got these pink cells are our new endoderm layer. Where do these cells come from? Well, these used to be up here. They used to be epiblast cells. Right? They went through here, they morphed into bottle cells and then to these mesenchymal cells. Mesenchymal cells, some of them, if they have high exposure to nodal, they go all the way down here to the hypoblast and kick out hypoblast cells and start to form a new layer called the endoderm. That is one of the germ layers. Mesoderm looks pretty well formed here already, right? That's another germ layer. So we got two germ layers formed, but we don't have, this is still epiblast. These are still, I mean, some of these are migrating cells, but a lot of them are just epithelial cells. So we got to fix that. So after the mesoderm is formed and the endoderm is formed, everything stops. No more migration, no more diving. And then something, we don't know for sure what it is, but something induces these primitive cells, these epiblasts, to morph into ectoderm, which is the third germ layer. Okay, that's pretty much the story of it. Mesoderm first or endoderm first. So there's a lot of confusion on this. It's agreed upon that the epiblast-derived mesenchymal cells are the ones that create the mesoderm, but we don't know which one forms first. We're going to go with mesoderm first. We'll go with more on that. Others say it's the endoderm first, like Larson, and others say it happens together. It probably happens together, but we'll just go with the board book on that one. Mesenchymal cells convert back to epithelial cells. Interestingly, the mesenchymal cells, yeah, it's weird. All that energy and work, and so all these cells finally get in here. Here's our new endoderm cells, and they're no longer stem cells, or they're no longer mesoderm. Once they get here, they morph back into an epithelial cell. So that's kind of weird. All right, so that has a name. So you could say that the cells of the endoderm have went through epithelial to mesenchymal to epithelial conversion, because they actually convert back into epithelial cells. That's not true of the mesoderm. The mesoderm does not turn back into endoderm, or does not turn back into epithelium. The mesoderm stays stem cellish, right? It's mesentery. It's not mesentery. Oh my God, my brain's fried from this week. They're mesenchymal cells, and they stay that way. Not forever, though. All right, here's just a couple of different, I think this is Larson, where we got cells migrating down. Larson says that here's the mesenchymal cells coming down and kicking the native, here's the native epiblast or hypoblast cells. It's just giving them the boot right out of there. And then day 16, it says then the mesoderm is filled in. We're not going to go with that, but if you're in Australia or wherever you're listening to this lecture, if Larson is your board reference book, then go by this. Fun facts: should be noted that the ectoderm forms in the cranial region first. Okay, that's fun fact. So that means that gastrulation actually proceeds in a cranial to caudal direction. Yes, that is true. Cranial to caudal. Primitive streak grows in which direction? That's a great little question. Cranial to caudal or caudal to cranial? No, it forms caudally, so it has the streak grows from the caudal region to the cranial region. And again, it's so important to understand that all of these germ layers are originally who's their daddy? The original cell, the epiblast cell, the pluripotent epiblast cells. All right, prechordal plate. Should have told this because this is kind of backing up a little, but nevertheless, let's tell it now. So prechordal plate, head organizer. Carlson calls it the head organizer because it organizes the brain and it forms the forebrain. We still don't completely understand it. Authors are all over the place with regard to its formation. Board books are still confused. Moore, chiropractic board book, says it's a thickening of the hypoblast. It's not a thickening of the hypoblast. Langman and Carlson basically are on the same page. It's really a collection of those mesenchymal cells, those mesodermal mesenchymal cells, that moves forward and forms a plate between the epiblast and hypoblast. So Moore, we're not going with you. Hopefully, I haven't seen, I don't know how they're going to handle that at the boards when you got conflicts like that. Prechordal plate, that is mesoderm built, we said. The mesenchymal cells are programmed, some of them are programmed to migrate straight forward. Some get exposed to high levels of nodal and go down and kick out the hypoblast cells, but these guys get special programming and they go straight forward in the midline and start to pile up. They go through the primitive node first. And just before an area that will become the oropharyngeal membrane, that's going to become the mouth. So it's very close to where the mouth is. And that pileup of cells is the prechordal plate. Carlson calls it the head organizer. We said that. So that was the first wave of cells that migrate straight forward. A second wave we'll talk about next time. A second wave of cells will become the notochord. I was thinking we're going to talk about it Thursday, but this is only once a week, this class. So here's a cool little picture. You can see the primitive streak here, and this is a mid-sagittal cut right through the midline. You can see the mesoderm starting to form here. This is the endoderm. Maybe it's still a little hypoblast, but regardless, we have a cell type coming through the primitive node and going straight up in the midline. And they're piling up into a little plate here, and that's called the prechordal plate, which is super important for forming the forebrain of the human. It forms right underneath the anterior visceral endoderm. Here's an overhead view of it. You can see the streak. So cells coming out of the primitive node go straight in the midline, and they pile up and form this prechordal plate. Okay, and while this is going on, that other story is going on. We got cells forming mesoderm underneath this layer. That would be underneath. We're looking at the dorsal part. We got cells doing this, in addition to going straight forward. So very busy. Prechordal plate. What does it do? Who's that little guy? That's the sonic hedgehog. And there's a family of genes called sonic hedgehog. We'll try to simplify it. So it turns out prechordal plate is a very important signaling center and induces, what's that mean, induce? It forms the early forebrain. So cells have to go through sonic hedgehog, or cells here have to go, or have to have the sonic hedgehog gene turned on. So in order to be these guys, your sonic hedgehog genes are all going to be firing. Okay? So what? Well, later, new mesenchymal cells will migrate through the prechordal plate, and as they pass through the plate, sonic hedgehog gene products tell them where to go. So you have to have sonic hedgehog turned on if you're a prechordal plate. All these cells have sonic hedgehog turned on. And so what? Doesn't do anything yet, but when cells come through here again and go further up this way, the sonic hedgehog will tell them, okay, you guys are going to become forebrain. Okay. Yeah, everything we said. Oropharyngeal membrane. You know what? I think that's probably good, right? I think that's enough for your brains. That's how many slides is that? That's like 70 slides. That's enough slides. Yep. Let's stop right there. All right. See you in the next video.